Good evening, I'm Charles Schultz, and this is the Randomizer, and it is the 16th of May, 2022. Now, tonight's term is going to be something a little odd that most people aren't familiar with, thermolabile. And what does it mean? Obviously, with the term thermo in it, it has something to do about temperature. It's about molecules, molecules that break down very easily with the application of heat. And so we're going to see why this is important and what it means, and how this is important to life in general, what abilities it can give an organism. So quickly, I'm going to run through my spiel. I'd like people to go to schultzlaboratories.com or universesciencecenter.com, go to our page, become a member, sign up, explore the galleries, get your Oculus or other 3D headset out, walk around in there and experience the virtual world as a learning environment. Play with some of the interactive exhibits, walk around and look at the everything that's available. If you're an educator and you want to share some of your knowledge, we can set up a gallery to display your works in a digital classroom in the virtual space for you to teach anybody in the world, and it's a lot of fun. It's a very engaging environment, easy to work with, and any sort of content can be added. If you have a company and you need to train your employees on a certain skill like operating a machine or navigating a certain situation, we can set up a virtual gallery for that to act as a training aid as well. Any of you who are interested in listening to the Randomizer live and participating by bringing your questions and comments and ideas, please do so Mondays and Fridays at 6 p.m. Eastern Standard Time. Go to our website page, click through the Randomizer link, and at a few minutes before 6 p.m. Eastern, it will automatically conduct you to the site where you can join us live. And be sure to bring some good questions. No sports questions, all technology, please. Okay, that being said, let's go on with our show and talk about thermolabile molecules. So first off, a lot of molecules have the ability to detect light or heat in some manner. Molecules have the ability sometimes to detect other molecules or to react in certain ways. And it usually takes a certain complexity of molecule. Light-sensitive molecules such as rhodopsin found in your eye, the thing that gives you night vision, that's a really well-known and well-understood phenomenon. Some molecules can absorb light and do things with it, such as photosynthesis in plants, producing starches and sugars. And that's an interesting way of converting light energy, which is essentially free, into something that plants and animals can live on. And when we look at other sorts of molecules, however, we find that their sensitivities to light or heat can have other interesting properties. Now, before I get into thermolabile, I want to talk about a light-sensitive molecule in a plant that will sort of bring you up to speed about why this is important. A lot of plants, as you know, will track the sun as it moves across the sky. You will see flowers or plants literally bend to follow the source of light. And you wonder, how does a plant without a brain, it's almost insensate, how does it know how to follow the light? It turns out that there is a class of molecules called brelins, in particular the most well-known gibberellic acid, which are actually light sensitive in an interesting way. Now, these brelin compounds act as growth enhancers or growth regulation hormones in many plants. And when exposed to light, they break down. They become ineffective. So a plant, when it's planted in the dark, and you may have seen this when you look into a bag of, or the bottom of the refrigerator, something has sprouted and it's tall, thin, and yellow. You look under a stairway outside by a porch, and there's tall, thin plants under there that are yellow. Why do they grow so tall and thin in the darkness? It's because plant cells produce brelin compounds. These compounds stimulate the growth of the plant, but at a cost. It takes energy. The idea is the plant will grow until it reaches a source of light, which is its power source. So the plant has these molecules that stimulate growth. But here's an interesting side effect. Because those molecules are light sensitive and will break down when exposed to a little too much light, they have an interesting effect on the growth and pattern of a plant. Suppose you have a stem, and on one side of the stem is a source of sunlight. The other side is the stem is in shadow. Obviously, a light-sensitive molecule on the sunlit side will break down and be ineffective. On the dark side, the molecule will be affected because it's not breaking down or not breaking down as rapidly. So here is the result. On that stem, the dark side of the stem will grow taller, the bright side of the stem will not grow, and the stem will curve in the direction of the light. So these molecules will literally stimulate the growth on the unilluminated side of the plant or the darker side of the plant, just enough to aim the plant directly at the source of light. At that point, there's a balance and no growth is necessary. As the sun moves across the sky, the plant's growth changes to follow the position of the sun, because where there's the most light, there is the least growth. And where there is the most shadow, there is the most growth. So this automatically orients the plant toward the sun. This is a photolabile molecule. So light destroys brelins. Brelins regulate growth. As a result, you see how the flower or the plant will track the movement of the sun over the course of the day. Now, thermolabile molecules, what would they do? Well, obviously, if it was in something that caused heat on one side or sensed heat on one side and not the other, and it made growth or a change of color, you could see that pretty obviously. But this isn't the case at all. In nature, many animals produce toxins or venom. And in the case of, let's say, cobra venom, this is a protein-based toxin. And interestingly enough, cobra venom is lecithin, the emulsifier molecule that is harmless, but 3 lecithins all put together and joined by a nitrogen atom. This triple lecithin molecule turns out to be a deadly poison. So it's easy to synthesize, and that's exactly what some set of genes in the cobra does. This is a protein-based toxin, and it's pretty durable. But some toxins are not made of proteins, they're made of other molecules. Wasp or hornet venom, bee venom, jellyfish venom, all of these contain thermolabile molecules. Now the term technically means any molecule that is easily broken down by a temperature of about 55 degrees centigrade or about 130 degrees Fahrenheit. But the fact is, even at lower elevated temperatures, it will accelerate the breakdown of this molecule. So it won't break down instantly, but over a brief period of time, if you apply enough heat, it will destroy these molecules. It's almost as if having a fever were the cure for destroying some microbes in your body. In a similar way, a high enough temperature can destroy certain toxins. So, I'm going to tell you a funny story, and I'm full of funny stories, but that's another story. When I was out with some friends performing a certain charity event about a year ago, one of the women who was going to perform in the event had 3 hornet stings simultaneously on one leg, and it was so painful she was about to the point where she could not perform. And I remembered about thermolabile molecules. I said, get a metal spoon, heat it up in some hot water until it's just barely tolerable. It's just as hot as you can take it, and put it on those areas where the stings are. And she did that. And within a few minutes, the venom had been destroyed. Now, it was uncomfortable, temperature-wise, but it destroyed the venom completely and removed the sting, the burning, and the pain. And she was perfectly fine. There used to be a device that was sold specifically for this purpose, and it looked like a pen, and it had a battery pack in it, which you could charge. And when you got stung, it could be, well, you press the button, and the tip would heat up, and it could be used to apply heat to the local area of the bite or sting and destroy the venom. So I don't know where these products are today, but they're actually quite useful. And for many people who are extremely sensitive to bee or wasp venom, they'll go into something known as anaphylactic shock. They will usually carry a syringe that they can inject themselves with that will help them fight this off, but they also often will have this small pen-like device to apply to the wound and destroy, using heat, the thermolabile toxins. So, and again, most toxins do not respond to this, but bees, hornets, wasps, and jellyfish toxins usually do. So if you're at the beach, you get stung by a jellyfish, you're in agony, heat up something, place it against the flesh, as hot as you can stand, and take a little time with it, and it will break down the toxin in almost every case, and it will alleviate that pain for you. So you don't have to suffer. So thermolabile molecules are molecules that have this ability. And why would that be necessary? Well, in some cases, organisms do need the ability to halt growth in an area where it's too hot, or to have other results as well. Molecules are actually messages for cells, and in many cases, the cell uses it as a message to grow or change course or produce another molecule or change color or do something. Most of the molecules in our environment are things that cells normally encounter and know how to deal with, and they can produce well-known effects. We use all sorts of molecules to change the growth pattern of plants, insects, your lawn, it doesn't matter. And in a sense, a fertilizer can be, but that's not really what we're talking about. That's a building block, and an essential building block for plant life. But in this case, here is a molecule that literally acts as a message or a switch, does something with a cell. It isn't necessary all the time to know what those things do, but simply to observe that a harmful effect can often occur, to know that you need to avoid that sort of thing. One of the problems that occurs in a modern industrial society such as our own is many times we produce chemicals that we use for certain jobs in the environment, such as pest control, and we don't know what the long-term effects are. And in many cases, nature does not have a mechanism of breaking these molecules down. One of them is the defoliant 2,4-D, which is also known as Agent Orange. Years ago, there was a microbiologist by the name of Chakrabarty, and he decided that it would be interesting to see if he could do a controlled evolution experiment where he could sort of force the evolution of microbes to see if they could tolerate 2,4-D. And so he started with some specific microbes, and he put it in a growth medium, and he cultured the microbes, and then he produced a zone where there was a small percentage of 2,4-D in the medium. And he watched the growth of the bacteria where it hit the medium and they stopped. But then within a few days, a curious thing happened. Some of the bacteria began to grow again and into the area with a small percentage of 2,4-D. So then he took those particular bacteria and cultured them with a concentration of 2,4-D, and he added a little more. And most of them died, but some of them survived to go to the next zone, which had like 5 or 10% 2,4-D in it. And this continued, but he also took another tactic. He used mutagens, chemicals that would induce mutations, such as retinoic acid and other compounds. By adding these compounds or exposing the bugs to a little low-level radiation or whatever, it would sort of throw the dice a little and cause a mutation. In this case, it was a mutation of some sort that allowed a higher tolerance to 2,4-D, the defoliant. Well, to make a long story short, after a couple of years of work, he had arrived at a species and evolved, a artificially evolved species of bacteria that could live solely on 2,4-D. It literally could eat it. So this is the power of evolution and the genetics that stands within every cell. You literally can make evolution occur on your own under proper circumstances. If you think that this is something strange or unusual, then consider, when's the last time you were out in the wild and saw a squash or a tomato or a turnip? For the most part, these things do not exist in the wild. They were artificially bred by human beings. Most of our food crops do very poorly in the wild because they don't have the genes to survive in the wild. They are cultured organisms that don't exist anywhere else in the world and just wouldn't make it. They're not tough enough. We've created huge artificial environments for these crops to grow in, and this is the source of our food. Many people are unaware that Arabidopsis, the common mustard plant, is the source of at least 5 of our vegetables today. Cabbages, Brussels sprouts, kohlrabi, it goes on and on. We have all of these, cauliflower and broccoli Broccoli and cauliflower are nearly identical, and sometimes you see crossbreeds of the two. All of them came by breeding the mustard plant for specific characteristics. So, if anybody asks you if evolution is real or if it exists or they express doubt, understand that there are many people in the world who not only see how it works, we see it around us all the time. They embrace it and they actually use it as a tool. I have used it as a tool for years, writing software, learning algorithms, developing new things, and it works wonderfully well. So if it's something that doesn't work, I don't see how that could possibly be. It's a demonstrated fact that it does work, and we see it in nature all the time. Well, without dragging that out, there are many cases, wallabies and other things that we see that literally have evolved to match an environment they've been imported into within a space of 7 or 8 decades. Um, we know that it happens, but thermolabile, that's the term at hand, and I should get back on the topic. You can see how, however, a single term can take you in many different directions and how many things are connected together. Thermolabile compounds, those that act as a functional tool or a message to a cell and can be destroyed by application of heat. Um, it may be something worth looking up. Do a little research and see what you'll find. Now, the floor is open for questions if anybody has any, and I'd be happy to take them. Meanwhile, I'm going to encourage you once again to become a member of Schultz Laboratories or the Universe Science Center. Support the development and growth of our galleries and exhibits. Our virtual world is growing, becoming more effective as an educational tool. And join us for the Randomizer, 6 p.m. Eastern Standard Time on Mondays and Fridays. And our robotics classes, Monday night, we have classes, and tonight will be lesson 6 of our robotics class. So be there, learn something cool, have a lot of fun with it. Well, I don't see any questions on the floor, and I'm going to read my quote for the day. We always have a random Bible quote. This one is, "Charm is deceitful and beauty is passing, but a woman who fears the Lord, she shall be praised." And that's Proverbs 31:30. All right. Well, I hope everybody found something interesting in my little expose here, and we'll look at thermolabile molecules and Brelin compounds, growth in plants and how they track the sun. Thermolabiles, as I mentioned, toxins from wasps, bees, hornets, and jellyfish can be destroyed by the application of heat in many cases. And that could be a helpful tool. It may be something you can use to help a friend in the future or even yourself. Well, I don't see any questions. It's been a lot of fun, and I hope that you have a wonderful evening. Take care of yourselves, be good to everybody, and as usual, make the world a better place. Talk to you next time.